37 degrees Celsius, 98.6 Fahrenheit. You've been that warm since the moment you were born. You know the number. Your doctor knows the number. Every nurse, every parent, every thermometer on the planet is calibrated around that number. But here is something I find remarkable. Not one person in 1,000 has ever stopped to ask why that number and not some other. Why not 40 degrees? Why not 30? Why not 25 or 50? Is it random? Is it arbitrary? Did evolution just land on 37 the way you might land on a channel while flipping through stations? It is none of those things. That number, the temperature inside your body right now, is the answer to a math problem. A thermodynamic optimization problem with three competing constraints, pulling in three different directions, and 37 degrees is the only temperature that satisfies all three simultaneously. One of those constraints is pure chemistry, one of them is structural engineering at the molecular scale. And the third, well, the third has to do with something that happened 66 million years ago, when the surface of the Earth was carpeted with fungus. I'll come back to that. And once you see the optimization, you'll understand something else too. You'll understand why your body sometimes deliberately breaks it. Why a fever isn't a malfunction. It's a weapon. Your body choosing to overspend because the threat is worth the cost. But let's start with the chemistry. Because the first thing you need to understand is that your body wants to be hotter than it is. You see, there's an equation in chemistry called the Arrhenius equation. It was written down in 1889 by a Swedish chemist named Svante Arrhenius, and it describes something deceptively simple. It tells you how fast a chemical reaction goes at a given temperature. The equation looks like this in plain language. The rate of a reaction depends exponentially on temperature, not linearly, exponentially, which means a small increase in temperature produces a large increase in speed. The reason for this goes back to something more fundamental, something Ludwig Boltzmann figured out in the 1870s. Boltzmann was an Austrian physicist, a deeply passionate man who spent his career fighting to prove that atoms were real. Most of the scientific establishment of his era thought atoms were a convenient fiction, a mathematical trick. Boltzmann insisted they were physical reality, and he built an entire statistical framework to describe how they behave. One of the things Boltzmann showed is that in any collection of molecules at a given temperature, the molecules don't all have the same energy. Some are sluggish, some are racing. The distribution of their energies follows a specific curve, and the shape of that curve depends on temperature. As temperature rises, the whole distribution shifts. More molecules end up in the high-energy tail. More of them have enough energy to overcome whatever barrier stands between them and a chemical reaction. That barrier is called the activation energy. Think of it like a hill. Every chemical reaction in your body requires molecules to climb over a hill before they can proceed. At low temperatures, most molecules don't have enough energy to get over the hill. They bump into each other, nothing happens, they wander away. At higher temperatures, more of them clear the hill. The reaction speeds up. The rule of thumb is striking. For most biological reactions, raising the temperature by 10 degrees roughly doubles the reaction rate. Double, from a 10-degree change. This is not a gentle increase. This is exponential acceleration. Now think about what this means for a living body. Every single thing you do, every thought, every heartbeat, every nerve impulse, every molecule of glucose broken down for energy runs on enzyme-catalyzed chemical reactions, thousands of them, tens of thousands, all happening simultaneously, all dependent on temperature. If you could run hotter, every one of those reactions would run faster. Your metabolism would accelerate, your muscles would contract faster, your neurons would fire quicker, your immune cells would divide more rapidly. From the perspective of pure chemistry, the Arrhenius equation has a clear preference. Hotter is better, always, no ceiling. The faster your chemistry runs, the more alive you are. If chemistry were the only constraint, evolution would have pushed body temperature as high as physically possible. You'd be running at 60 degrees, 70 degrees, as hot as the machinery could tolerate. But chemistry is not the only constraint. And this is where the second force enters the picture. The force that pushes in exactly the opposite direction. Every enzyme in your body is a protein. And a protein is not a rigid little machine. It's a long chain of amino acids folded into an extraordinarily specific three-dimensional shape. The shape matters. The shape is everything. An enzyme works because its shape creates a precise pocket where a specific molecule fits, where a specific reaction is catalyzed. Change the shape, and the enzyme stops working. It's like crumpling a key. The metal is still there, but it no longer opens the lock. What holds that shape together? Not covalent bonds, not the strong chemical bonds that hold atoms together within molecules. The three-dimensional fold of a protein is maintained by much weaker forces. Hydrogen bonds, van der Waals interactions, hydrophobic effects. These are the gentle forces, the ones that can be disrupted by modest amounts of energy. And temperature is energy. As you raise the temperature, those weak bonds start to vibrate more violently. The protein begins to unfold. Slowly at first, around the edges, the less stable regions losing their shape, then more rapidly. And somewhere between 40 and 45 degrees Celsius, for most human proteins, the unfolding becomes catastrophic. The protein loses its functional shape entirely. Biochemists call this denaturation. The protein is denatured. It's the same thing that happens when you fry an egg. The clear albumin turns white and solid. That's protein denaturation. The molecules have unfolded and tangled into a useless mess. It's irreversible. Think about that. The difference between a working enzyme and a useless tangle of amino acids is about 6 degrees. Your operating temperature is 37. Your proteins start to fail at 43. That is your entire margin. 6 degrees Celsius. In engineering terms, that is a terrifyingly thin safety margin. A bridge designed with that kind of tolerance would make engineers nervous. Your body knows this margin exists. At 42 degrees, before full denaturation sets in, your cells activate an emergency system. They start producing what are called heat shock proteins. These are molecular chaperones, proteins whose entire job is to grab other proteins that are starting to unfold and physically hold them in the correct shape. They're the emergency crews rushing to reinforce a structure before it collapses. The fact that your body has an entire emergency protein system dedicated to this tells you something important about how close to the edge you operate. You wouldn't build a fire brigade if fires never happened. The brigade exists because the danger is real and constant. So now you have two forces. The Arrhenius equation pulling you hotter, always hotter, demanding faster chemistry, and protein stability pulling you cooler, insisting that you stay below the denaturation threshold or everything falls apart. These two forces are in direct opposition. Here is where it gets interesting. If you graph the first force, enzyme efficiency as a function of temperature, you get a curve that climbs steeply. More heat, more speed, more throughput. If you graph the second force, protein structural integrity as a function of temperature, you get a curve that's flat for a while and then drops off a cliff somewhere around 40 to 45 degrees. Now multiply those two curves together. The product, the combined curve of how well can your body actually function at this temperature, has a peak. Below the peak, you're leaving performance on the table. Your chemistry could run faster, but it doesn't, because you're too cold. Above the peak, you're destroying the machinery. Your reactions are faster, but your enzymes are falling apart, so the net output drops. The peak of the product curve sits at approximately 37 degrees Celsius, not because anyone designed it to be there, not because some cosmic engineer picked a nice round number. It's there because of the mathematics. Given the activation energies of human enzymes, given the thermal stability of human proteins, the point of maximum net function falls at 37 degrees. Your body temperature is the solution to an optimization problem, the only temperature where you get the most chemistry without breaking the hardware. And for a while, that seemed like the complete answer. Two constraints. One, optimization. A satisfying explanation. But I told you there were three constraints, not two. And the third one is the strangest of all. It has nothing to do with your enzymes, nothing to do with your proteins, it has to do with fungi. In 2010, two researchers at Albert Einstein College of Medicine published a paper that changed how biologists think about body temperature. Their names were Arturo Casadeval and Vincent Bergman. Casadeval was an infectious disease specialist, not a physicist, not a thermodynamicist. A doctor who spent his career studying fungal infections, and he had noticed something that puzzled him for years. Fungal infections devastate cold-blooded animals. Reptiles, amphibians, insects, they are constantly battling fungal pathogens. There's a fungal disease called chytridiomycosis that has been wiping out frog populations across entire continents. Another fungal pathogen has been decimating bat populations during hibernation when their body temperature drops. In insects, fungal infections are so common that some fungi have evolved to literally take over an insect's body, control its movements, and force it to climb to a high point before erupting with spores. Nature is saturated with fungal predators, but mammals? Mammals barely notice. Healthy mammals shrug off the overwhelming majority of environmental fungi. Fungal infections in humans are serious primarily for people with compromised immune systems. For a healthy person at normal body temperature, the fungal kingdom is almost entirely irrelevant as a threat. Casadeval wanted to know why, so he and Bergman did something clever. They compiled data on the thermal tolerance ranges of tens of thousands of fungal species from the environment. Every species has a maximum temperature at which it can grow. Plot all those species on a graph with temperature on one axis and the number of species that can survive at that temperature on the other, and you see something remarkable. Below about 25 or 30 degrees, the vast majority of fungal species thrive. But as you go above 30 degrees, the number of species capable of growing drops sharply. Roughly 6% fewer species for every 1 degree Celsius you add above 30. By 37 degrees, you've already eliminated the majority of environmental fungi. By 40 degrees, almost none can survive. Your body temperature sits right in the zone where most fungi simply cannot grow. You are too hot for them, not by accident, not as a side effect of metabolism. Your temperature is a thermal exclusion zone. But here's what makes the paper genuinely beautiful. Casadeval and Bergman didn't just observe this. They built a mathematical model. They asked a specific optimization question. If you're a warm-blooded animal, maintaining a constant body temperature costs metabolic energy. The hotter you run, the more food you need, the more oxygen you burn, the more energy you spend. That cost goes up with temperature. At the same time, the higher your temperature, the more fungal species you exclude from your body. The benefit goes up with temperature too, but the cost goes up faster at the high end, while the benefit starts to plateau because you've already excluded most fungi. When they optimized the model, when they found the temperature that gives you the maximum fungal protection per unit of metabolic cost, the number that came out was 36.7 degrees Celsius. Think about that for a second. A model that knows nothing about the Arrhenius equation, nothing about protein denaturation, nothing about enzyme kinetics. A model built entirely on fungal ecology and metabolic cost. And it produces the same temperature as the biochemical optimization, within a fraction of a degree of actual mammalian body temperature. Three completely independent physical constraints: chemistry demanding heat, proteins demanding caution, fungi demanding a thermal barrier. And all three converge on the same narrow window. That is not coincidence. That is convergence under physical law. And now the evolutionary story comes into focus. 66 million years ago, an asteroid roughly 10 kilometers across struck what is now the Yucatan Peninsula. The Chicxulub impact. The immediate devastation was catastrophic. Fires, tsunamis, a cloud of debris that blocked sunlight for months, possibly years. Temperatures plummeted, photosynthesis collapsed. But in the darkness and the decay, something thrived. Fungi. The planet's surface became a feast of dead and dying organic matter, and fungi exploded across the Earth. A fungal bloom of staggering scale. Now, consider who was alive during this catastrophe. The dinosaurs, or at least the non-avian dinosaurs, were primarily ectothermic, cold-blooded. Their body temperatures tracked the environment. In the cold, dark aftermath of the impact, their body temperatures dropped into the range where fungi thrive. They had no thermal barrier. They were vulnerable not just to starvation and cold, but to fungal infection at a scale the planet had never seen. And the mammals, small, warm-blooded, already running their bodies at 37 degrees, already sitting inside the thermal exclusion zone. The fungal bloom that covered the earth couldn't touch them, not because they had better immune systems, not because they were hiding, because they were too warm. This is the open loop I planted at the beginning, the catastrophe 66 million years ago, the surface of the earth carpeted with fungus, and the warm-blooded animals walking through it untouched. Your body temperature is not just a chemical optimization, it is a survival strategy that predates human civilization by tens of millions of years. You carry the legacy of that strategy in every degree of warmth you radiate right now. Now let me tell you about fever, because this is where the body does something that seems, on its face, irrational. It deliberately raises its own temperature. It moves off the optimum, away from the peak of the product curve, into the danger zone. When you get an infection, certain molecules called pyrogens, some released by the pathogen itself, some produced by your own immune cells, travel through your bloodstream to the hypothalamus. The hypothalamus is the body's thermostat. It's the region of the brain that maintains your temperature at 37 degrees by balancing heat production and heat loss. Shivering when you're cold, sweating when you're hot. A continuous, exquisitely sensitive feedback loop. Pyrogens reset the thermostat. They tell the hypothalamus that 37 is no longer the target. The new target might be 38.5 or 39, or, in severe cases, 40 or higher. And the hypothalamus responds exactly as it would if you were actually too cold. You shiver. Your blood vessels constrict. You feel chilled even as your core temperature climbs. You pile on blankets. Your body is generating heat as fast as it can to reach the new, higher set point. This is not a malfunction. It's a strategic decision. Every degree of fever costs approximately 13% more metabolic energy. That is an enormous expenditure. Your heart beats faster. You breathe more rapidly. You burn through glucose and fat reserves at an accelerated rate. This is why fever makes you feel exhausted, why you lose your appetite and feel drained. Your body is pouring resources into heat production at the expense of almost everything else. But look at what that extra heat buys. First, it accelerates your immune response. Your white blood cells divide faster at higher temperatures. The Arrhenius equation works for your immune cells too. Antibody production increases. Phagocytes, the cells that engulf and destroy pathogens, become more active. The chemical warfare your immune system wages runs faster and hits harder. Second, it shrinks the pathogen's survival range. Many bacteria have optimal growth temperatures near 37 degrees. They've evolved to thrive at exactly your normal body temperature because that's the environment they inhabit. Push the temperature to 39 or 40, and you moved away from their optimum. Their metabolism falters. Their reproduction slows. You've changed the battlefield. The body is doing a cost-benefit calculation. Under normal conditions, 37 degrees is optimal. The cost of running hotter isn't worth the marginal benefit. But when there's an active infection, when pathogens are multiplying inside you, the calculation changes. The cost of running hotter is still enormous, but the cost of not running hotter, of letting the infection win, is your life. So the body overspends. It accepts the 13% per degree surcharge because the alternative is worse. This is why suppressing a mild fever with medication is more complicated than most people think. You're not fixing a broken thermostat, you're overriding a deliberate tactical decision your body made. In many cases, the fever is helping. It's part of the defense. Of course, extremely high fevers are genuinely dangerous because you're pushing toward that denaturation cliff I described earlier. Above 41 or 42 degrees, the risk of protein damage starts to outweigh the immune benefit. The body normally prevents this, but sometimes the system overcorrects. That's when fever becomes a threat rather than a tool. Now I want to show you where all this heat actually comes from. Because you might think of body heat as something separate from the rest of your biology, some dedicated heating system running in the background. It's not. Your warmth is a byproduct, a leftover, the exhaust of being alive. Every cell in your body runs on a molecule called adenosine triphosphate, ATP. It is the universal energy currency of life. When you flex a muscle, when a neuron fires, when a cell divides, when your heart beats, the energy comes from breaking a phosphate bond on ATP. This is called ATP hydrolysis. The efficiency of this process is roughly 40%, which means for every unit of chemical energy stored in ATP, about 40% gets converted into useful work. The actual mechanical contraction of a muscle or the actual transport of an ion across a membrane. The other 60% is released as heat, waste heat. Thermodynamic exhaust. You produce roughly 80 watts of power at rest, about the same as an incandescent light bulb. Of those 80 watts, approximately 48 watts are waste heat. They serve no biological function. They are the thermodynamic cost of doing business. Every chemical reaction in your body that does useful work also generates heat as a side effect, because no energy conversion process is perfectly efficient. This is not a limitation of biology, it is a law of physics. The second law of thermodynamics guarantees that some energy will always be lost as heat in any conversion process. There is no workaround, there is no cheat code, inefficiencies baked into the structure of the universe. And here is the thing I find most beautiful about all of this. Evolution didn't fight the inefficiency. It couldn't. The laws of thermodynamics are not suggestions. Instead, evolution took the waste heat that is unavoidable, the 60% that was always going to be lost, and it built a defense system around it. Your warm-bloodedness is not a separate system from your metabolism, it is your metabolism. The heat that keeps you at 37 degrees is the same heat that's released when ATP is hydrolyzed to power your muscles, your brain, your organs. Evolution simply captured that waste heat, wrapped it in insulation, skin and fat and fur in other mammals, regulated it with a hypothalamic thermostat, and used it to maintain a body temperature that happens to sit at the precise intersection of three optimization curves. It is an act of extraordinary thermodynamic elegance. Not the elegance of efficiency, but the elegance of repurposing. Taking what physics gives you for free, the unavoidable waste product of every energy conversion, and turning it into a fungal barrier, an immune accelerator, and a chemical reactor running at peak throughput. You are an 80-watt light bulb that learned to weaponize its own waste heat. Let me bring you back to where we started, your hand on your neck. That warmth under your fingers, you felt it your whole life. You've never thought twice about it. It was just there, like the weight of your own body, like the sound of your own breathing. But that warmth is not incidental. It is not arbitrary. It is the solution to an equation, the peak of a curve, the legacy of an asteroid, and the exhaust of 10 trillion cells converting sugar into motion. It is the number where chemistry runs as fast as it can without breaking the machinery. It is the number where fungi cannot follow. It is the number where the cost of staying warm and the benefit of staying warm reach their optimal balance. 37 degrees Celsius. The temperature at which being alive works best, given the laws of this universe. You are not warm despite being alive. You are warm because being alive is thermodynamically inefficient. Every cell in your body loses 60% of its energy as heat, and evolution took that loss, that unavoidable physics-mandated loss, and turned it into armor, into a weapon, into the reason your species walked through a fungal apocalypse 66 million years ago and came out the other side. The warmth you feel right now, under your fingertips, against your neck, is the waste heat of 10 trillion molecular engines, captured and regulated and repurposed into a thermal fortress that has been running, uninterrupted, for the entire history of your life. And I think that is one of the most beautiful things physics has ever explained.